We're ready po. Dr. Sarah, we're ready po.
Okay. Good morning, good evening, ladies and gentlemen. Welcome to the PAASE for Philippine Innovation Fireside Chat series. And this is the fourth we are holding. And today we have a very special, interesting speaker who is an innovator in his field of expertise. And I would like to at this point introduce first my co-host. Many of you know him already. He's Dr. Alvin Colaba of the De La Salle University in Manila. And I think I don't need to mention his credentials anymore because he's very famous, except to say that he's a vice president of the National Academy of Science and Technology, former president of PAASE, former president of the NRCP, and a distinguished professor in DLSU, mechanical engineering. So let's flash the slide of our speaker today. Um, we're happy to have Dr. Homer Pantua, who will speak on revolutionizing animal health in the Philippines, biotech beginnings. And now I'd like to yield the floor to Dr. Kulaba to introduce our speaker. Thank you.
Thank you, Academician Giselle Concepcion. Of course, we know Giselle, almost all of us, all of you knows Giselle, but certainly she is currently an executive member of the National Innovation Council, a presidential appointment that she has to actually hold for the next 3 years. And I'm co-hosting with Giselle this series of our Fireside Chats on PAASE for Philippine Innovation. Today, on our fourth edition of our series, we have a distinguished veterinarian and scientist who has led both antiviral and antibacterial drug discovery efforts. A senior principal, scientific researcher, and laboratory operations manager at Genentech, a pioneer in the development of therapeutics from recombinant DNA technology. Our distinguished speaker obtained his PhD in biomedical sciences from the University of Massachusetts Medical School in the US and developed a patented technology on virus-like particles as a vaccine platform for human and animal pathogens. He received his Doctor of Veterinary Medicine from the University of the Philippines. Of course, this is in Los Baños, I suppose. He initially received infectious disease training at the Research Institute for Tropical Medicine. Currently the CEO of BioAssets Corporation, a pioneering life science company in the country, engaged in veterinary research and diagnostics. He is the leading company's research and development initiatives. Our speaker is a project leader of a DOST-supported initiative under the Business Innovation for Science and Technology, or BIST, which aims to address the unmet needs in the animal industry. Currently involved as a technical advisor in the establishment of the proposed Virology and Vaccine Institute of the Philippines. Ladies and gentlemen, it is my pleasure and honor to introduce our speaker, Dr. Homer Pantua. Homer, the floor is yours.
Yeah, thank you very much, Dr. Alvin Colaba and Dr. Gisela Concepcion. Thank you to my fellow PAASE colleagues and to all the attendees. Good morning to all of you. It's a pleasure to be here. Can you see my slides? Oh.
Yes.
Yeah, so as mentioned today, I'm going to be talking about our efforts in revolutionizing animal health in the Philippines and the biotech beginnings of BioAssets. And today I'm going to give you a short overview of our company and what's the unmet need in the animal industry that we are addressing, and our research and development activities. And I'd also like to introduce to you the BioAssets Academy. So BioAssets is a pioneering life science company. It's a 100% Filipino-owned and also an employee-owned company in the Philippines, which is established by a team of scientists, veterinarians, farmers, and entrepreneurs based on the foundation of serving the people. So our ultimate objective is to contribute to the advancement of food security in the Philippines. And we firmly believe that a sustainable and thriving animal industry is necessary to support food security in our country. So whenever I give a talk, I always introduce our people and our core values, because this is the center of what we do and how we succeed in our ventures. So BioAssets activity is backed by a really good team of people, which is guided by our core values. So our first core value is truth seeking. So our company follows the science. So whatever the science says, we follow. If the science says no, we stop. If the science says go, we proceed. So we are a purpose-driven company. We are focused on the projects. We are focused on the goals. So we are a company that we have a genuine care for people, animals, and the environment. And we believe that we cannot do anything on ourselves. That's why we value collaboration. And we are a company who invests in people, because we believe that investing in our people to reach their full potential, it's not only beneficial to our company, but most importantly to the industry that we serve. And we are a company that promotes innovation culture. So with these people and these core values, we have established partnerships and collaborations here and abroad, for example, in the United States. And most notably, we have a strong collaboration with Kansas State University, working on the African swine fever virus also. And we are also establishing one in California with UC Davis. And we also have commercial partnerships and research partnerships in the Europe and also in Asia, such as Thailand and Malaysia, and a very strong collaboration with an animal productivity enhancement in Australia. So in these areas, our operational activities are actually encompasses disease diagnostics. In 2014, we are the youngest and the youngest in the field of commercial diagnostics. But now the animal industry have considered BioAssets as the largest commercial diagnostic laboratory in terms of market share and in terms of capabilities. But we did not, our objective is not to be a disease diagnostic company only, but we want to be an R&D company that develop products and systems. So that's why we invested in genomics. We also invested in molecular food micro. We also established information management system for our clients, such as corporate farms and other biological companies. We also do biosamples management to support our vaccine development program. And one of the problems here in the Philippines is importation of reagents. That's why we're also venturing into that. Actually, it's going on right now. And we position BioAssets as a contract research organization where we actually do research planning, execution, and management with other biological companies. We also do technology commercialization with other companies here and abroad. And some of the research and development activities that we do at BioAssets is that we've developed point-of-need diagnostics platform, and which I will be talking briefly with you. We also have initiatives on customized vaccine or autogenous vaccine. We're also looking into establishing lab animal medicine to position ourselves as one of the contract research organization, not only in the Philippines, but hopefully in Southeast Asia. We're also venturing, we have R&D activities on food biological, for example, recombinant DNA production of proteins that are needed in the industry. We also have enhancement of breeding using boar semen sorting, which I will discuss with you today. And as I've said earlier, one of the problems of doing research here in the Philippines is procurement of reagents, and almost all to the simplest of water is imported in the Philippines. That's why we are actually excited to do this, and we are actually putting this center at the UPLB APEC, which is a PESA area. So now I'm going to give you an overview of the animal industry outlook. And in the next 3 years, from 2018 to 2016, the animal health business has been forecasted to have 6.1% growth. And it is driven by surge in population, increasing demand for high-value animal protein, and also increasing demand for companion animals such as dogs and cats. However, the emergence of infectious diseases negatively impacted this growth, especially with the case of the African swine fever virus and the avian influenza. So right now, infectious diseases still remains the major cause of losses in animal production. And until now, despite the technological advances globally, our country still lacks the ability to rapidly respond to outbreak at the point of need. And we also, even though we have some capability, we still have the lack of comprehensive molecular characterization of pathogens that are present in the Philippines. And right now, we still don't have safe and effective vaccines for many diseases. So those are the things that BioAssets would like to address. And also in our country, the way we approach animal health is very reactive. So usually farmers and veterinarians seek for diagnostic when they see clinical signs. And in most cases, when you see clinical signs, it's too late already. Basically, what happens is just whatever diagnostics that we do, it's basically a damage control, which equates to cost. And some of the things that we do is only necropsy, histopath, and serology because of cost. So, but what we advocate at BioAssets is that we need to be proactive in our approach to animal health. We look at the animals and we also look at the environment. And we have to dig deeper. Other than serology, we have to look at the definitive diagnostics such as PCR diagnostics, and we have to look deeper also on the genomics of the pathogens. So in that way, we can actually institute preventive, which equates to insurance, and that means diagnostic can give meaningful information. It can also give value of information. That value of information offsets the cost of diagnostics, and in that way, the information will support sound decision-making. So I'd like to introduce to you one of our projects, the Bridges project, which is supported by the Department of Science and Technology under the Business Innovation for Science and Technology under the Science for Change program. So Bridges is a bridge response for in-location diagnostics and genome sequencing. And the objective of the project is to establish a comprehensive and unified approach to address infectious diseases in animal industry. And the aim of this project are focused on 3 areas, such as point-of-need diagnostics, where we can establish our capability to respond at the point of need, precision diagnostics. And these 2 things, point-of-need diagnostics, precision diagnostics, will actually set us up for precision medicine, wherein we can actually make customized vaccines for the animal industry. So right now, we've established the very first mobile biocontainment laboratory, which is deployed in Central Mindanao University. With this platform, we were able to establish rapid response to outbreak. We are actually working in the prototyping now with farmers and commercial farms, backyard farmers and commercial farms. Through this, we can actually support continuity of operation, sound decision-making for veterinarians and farmers. It also support capacity building, and it can also support research and self-surveillance while maintaining biosafety. And we are very, very proud and glad to share with you that this platform has been granted by the health security partners a challenge grant to further develop the system that can help the Philippine animal industry. So this platform will also be showcased in the National Science Technology Week in November in Iloilo. So we have proven that through this platform, we can actually do on-site testing, on-site analysis in a timely and high throughput manner. In such a way, we can help the farmers make sound decisions at the point of need. So the idea is for the Department of Agriculture and local government units to adapt and implement the program, but it's not enough. So we have to continue the innovation. That's why we are partnering with state universities and colleges. There are College of Veterinary Medicines. There are 18 in the Philippines. So The idea is to establish training centers and R&D hub such that we can continue the innovation and technology transfer through generations. So that's with the point of need diagnostics. But in just under 2 years of the Bridges project, we have established precision diagnostic collaboration with many different collaborators in the academia here and abroad. And one of the most notable collaboration that we have is in African swine fever virus. And just to let everyone know, BioAssets is the very first company who have generated the very first locally sequenced and assembled African swine fever virus. And we are continuing to do all the surveillance and genomic studies on African swine fever virus such that we can actually help give information that are important for diagnostic development and vaccine research. We are also working on avian influenza in collaboration with another Balik scientist, the Philippine College of Poultry Practitioners, UPLB, UP Diliman, and DOST. And hopefully in the very, very near future, very near, like in the next 2 weeks, we will be launching the very first whole genome sequence of avian influenza that is locally generated and assembled. So other collaborations that we have on bacteriophages for industrial purposes, salmonella for food safety, and Proteus millabilis for other with the National Pingtung University of Science and Technology. And we are open for collaborations. We support people, we support graduate students, and what we hope is to support an explosion of information such that we can use that to address unmet needs here in the Philippines. So one of the unmet needs that I mentioned earlier is the lack of safe and effective vaccines. And in some cases, there are available vaccines, but pathogens evolve and adapt to selection pressure. That's why it results to vaccination failure. And what we do at BioAssets is we use customized vaccine to support precision vaccinology for the animal industry. What we do is we identify sick animals and we do comprehensive diagnostics of these sick animals. So what I'm going to do today is I'm just going to give you an overview of what we do. We're not going to go deeper to the technology, but here, after comprehensive diagnosis, we do isolation and biosamples management of these bacteria and viruses in such a way that when there is an emergency need for vaccines for a swine farm or a poultry farm, we can generate vaccine right from the isolates or through recombinant DNA technology. So one of the things that we do is also for productivity enhancement of us as the swine breeders. So because of the ASF, the devastation caused by ASF, there is a need for female breeders, and the industry right now is having hard time to cope up with the need for the female breeders for repopulation. And in a swine farming, the female breeder is very important for productivity. And how do we approach this need? So what we do is basically in any operations right now in commercial farms, breeding is actually done by artificial insemination. So what we think is that what if we can actually enhance for female spermatozoa such that we can increase the number of females in the offspring? So what we're doing right now in collaboration with our another scientist in Australia is we are co-developing an antibody that can actually deplete the male spermatozoa in such a way that we can actually enrich female spermatozoa in the AI sample, such that if we enrich the female spermatozoa, we can actually increase female offsprings in the litters. So what did we see in our initial experiments? So what we see in a treatment in a control group is that there is no difference with litter size. However, when you treat these female pigs with our antibody, the number of live pigs per insemination is actually better compared to the control. And the consumption rate reaches 100% compared to the standard acceptable consumption rate of close to 90%. And what's very interesting and very encouraging is that when we treat our sows and gilts with our antibody before insemination, we can actually enhance the ratio of male to female. So we can get 30 to 70% female. So that's very encouraging. So what I want to show you is the economics of female enrichment. So in a commercial production or a smallhold production, what we do is we either do natural or a standard AI, it's artificial insemination of our sows and gilts. And in naturally, there will be 50% female and 50% male. So in a litter of 12 pigs, you would expect six females. So what we expect is that when we use our antibody to do semen sorting and enhance the female, what we want to expect is to increase the female from actually 60% to 70% and the male to 30 to 40%. That's the reverse. So in a litter of 12, what we would expect now is from a 6 from the natural or standard AI, we will be expecting 8 females now. It looks like if you look at one sow, it doesn't matter, it's only 2. But if you look at the potential of this technology, assuming that you have 1,000 sow level, that would equate to 2,000 sows in the selection pool. At 2,000 sows in the selection pool will actually add another 50 million pesos in revenues for the breeders. So it's a very, very good, it's a very promising technology that we are actually doing here in the Philippines right now. So another thing that we do other than the R&D activities is we want to strengthen the government, academia, and industry linkages. And the way we do it is we look for government supports, research fundings. We're establishing research centers. Actually, right now we're going to establish a research center in UPLB at the APEC district. We're also establishing advisory boards, academic industry joint projects. Which I will be sharing with you right now. We also establish internship and training programs. We establish knowledge exchange platforms, and we put a lot of considerations with IP protection and tech transfer to foster trust and encourage collaboration. And for this to be successful, we have to continuously engage the government, academia, and industry to establish a robust life science atmosphere in the Philippines. So what are we doing right now? So right now, we have a government, academia, industry collaborations, such as these 3, actually 4. The first is we call it PREVENT. This project is conceptualized by BioAssets and another Balik Science in the name of Christina Layson and our colleagues at UPLB. And the idea is it's a pandemic response preparedness for emerging bat-borne viruses from rural and peri-urban environments. And the idea is to establish vaccine research and virus surveillance against potentially viruses that has potential, but that has pandemic potential. Another is on avian influenza, which is basically on rational vaccine using the epidemiology of avian influenza viruses in domestic and wild bird communities, again in collaboration with UPLB, UP Diliman, the ITDI, and the Philippine College of Poultry Practitioner. And also we have some initiatives with African swine fever. One is have gone so far already is the point of need PCR diagnostic kit, which is led by the Industrial Technology Development Institute. And we are also establishing a collaboration on developing nanoparticle DNA subunit vaccine in collaboration with Holland Cheng of UC Daisies. I think I saw him here. So it's very exciting times, and I'm very glad that the government is actually supporting this project. There is a really good, the outlook of life science and vaccine research in the Philippines is very promising. And I hope that we can actually, the government will continue to support it. And what we're doing right now as a company is other than our R&D activities, is that we serve as a driver and catalyst of the life science atmosphere here in the Philippines. And what we see is that we could continue to engage the government. We are a strong supporter of the Science for Change program. We have collaborations with academia, particularly University of the Philippines, Los Baños, and UP Diliman. We are very glad to share with you that BioAssets has now 4 PhD students, 3 PhD by research and 1 standard PhD. Really exciting times for science here in the Philippines in both academia and industry setting merging together. We also have collaboration with Research Institute, even in the life science industry and the key opinion leaders. Not just that, given our commitment to the future of science in the Philippines, we have established the BioAssets Academy. This is a venue for knowledge transfer, skills development, and professional empowerment. And up to now, we have trained many veterinarians and microbiologists to be industry ready. And we are glad, and this academy is open to all universities here in the Philippines and abroad. We've also established knowledge platforms for knowledge transfers and networking so we can foster collaboration and encourage innovation here in the Philippines. And I just want to share with you the roadmap of our company. Basically, we're putting the pieces together to reach success as a life science company. So we started as a diagnostic lab 9 years ago. We've developed skills, we've taken knowledges, and we've transferred knowledge to the other people. We've started surveillance programs such that we can establish our own biobank. We've also established on-site and precision diagnostics platforms to support our genomics. And we've also started translational research. We've invested in our people to learn how to do translational research. And one of the things that we want to do as a contract research organization is to establish a lab animal medicine. We are now in the groundwork of doing one. We've actually invited people, our collaborators from the United States who are experts in CRO. And also, ultimately, we want to be a company that produces vaccine for the animal industry. Hopefully, hopefully for the Philippines and Southeast Asia. With that, I would like to invite you to join us in nurturing life, science, and innovation to serve the people. Maraming salamat po.
Wow, it was great. Thank you very much, Homer. We are, there's a lot of hope, in the Philippines with the development of your BioAssets company here. Let me start not on the technical side, Homer. I think it's very clear, what you are doing for the animal industry. But you have been supported by a government program under DOST, the BIST. Hello? Giselle?
Yes, I'm here.
Okay. I am hearing something, So let me start, with this program of the government, which is the BIST. This is the business industry for science and technology as this, Homer? Is that correct?
Yeah, the business innovation for science and
Innovation
technology.
for science and technology. Yes. So how much, I would say, support that under this program allowed you to establish your company in this in the Philippines?
Yeah.
And what was it was it easy for you to access this grant, and so that at least we know, in other innovators who would like to do or to access similar program of DOST can probably consider that. Can you just share your experience in accessing that particular program, and how much the support has contributed to the success of your BioAssets?
Opo. Thank you very much for the question po. I'll give you a little bit of history, and I would like to acknowledge one of our PAASE colleague who actually advocated and introduced us to USA Stride. It was actually the USA Stride who recommended us to DOST BIST. So BIST is actually part of the Science for Change program. It's designed for companies older, 5 years and older. And when I met our PAASE colleague, Dr. Al Serafika, he was excited about the technologies, and he said, "Oh, this might be good for you." And that it started from that. And then what happens is that we applied for the grant. Basically it's a grant through DOST. The maximum grant, it's not a grant actually, it's a soft loan without interest. The project is 7 years. 2 years, you have a grace period. You start paying on the 3rd year. The maximum amount of money that you can get is around 75 million with no interest, payable in 5 years, starting on the 3rd year. And BioAssets was granted 15.9 million to support the Bridges project. And basically, but we have to have a counterpart too, which is around 20% of the project in forms of personal services and operational expenses. So, but I think it's a really good program. It's basically a grant writing activity. It's good, but what I'm happy about the program of the BIST and the Science for Change program is that everything happened in the pandemic, but you know what? The moment we got approved, we got the money in 2 weeks.
Oh, that's great. Maybe Homer, later on you can share, how others, can actually prepare a grant proposal for that kind of program.
Yeah, of course.
Yeah. We see here Engineer Rolly Sevilla raising his hand, and of course later on Al will call you after. Engineer Sevilla, Rolly.
Hi, good morning, Dr. Thank you so much, Engineer Dr. Homer. Good morning to everyone. I'm Engineer Rolly de Sevilla. I have 5 companies which is engaged in green renewable energy, fintech solutions, corporations. Actually, we are into one with the innovation, the launching of the national agenda for innovation and strategy. So right now we are designing the platform for LRA as well as with the local government units, so that the business permits, the processing of doing business in our country will be easier. Okay. My question is, because I would like to make an investment with food security, as well as with, of course, I'm also into rice and sugar export imports. But I'm looking for to invest in poultry or in animal husbandry, like cows and what do you call this, goats. Okay. What I'm trying to ask to Dr. Homer is, I knew that our animal husbandry, specifically cows, is not internationally competitive.
Yes.
Because the cows are coming from Australia, New Zealand. So how what I'm thinking is, if ever I'm going to invest here, I would like to have someone to make the cows, considering our climate. Okay, how we can make our cows to be their meats is tender, like like the quality, almost like the quality of New Zealand, cows from New Zealand, beef from New Zealand, and Australia as well. Do you have any plans to help the animal husbandry industry in these terms?
Yeah, of course, po. Right now we don't have a program on cows, but if you go back to the Boris or semen testing that I shared with you, that's we also have that in bulls, but it's not for like to make it tender or some sort of a better meat quality. We're only on the production side. We want to increase the number of females such that there will be more dairy cows. But of course, if there is a need for that, if we can, for example, if there is a need for molecules or any proteins that we can help, or we can definitely talk, and we can definitely establish something or explore something such that we can use science to explore those questions.
Yes, yes. I'm really grateful to be connected with you through this PAASE for Philippine Innovation, because I have 1,200 hectares in Gabaldon, Nueva Ecija, and some parts of the property I would like for our food security, because we're looking forward, because our national security is actually threatened right now by other countries, which specifically China. They are already invading the West Philippine Sea. They just recently they just make a artificial island near Zambales. So we are really looking to have a food security. So I'm really grateful to have a collaboration with you. If ever this platform can make us connected, I will highly appreciate it. Thank you so much. Have a great day.
It will be my pleasure, po. We would be glad to help you, po.
Thank you, Engineer Sevilla. We'll be in touch with you.
So
Of course.
Alvin,
Yes, Giselle. Now,
yes. So
Go ahead.
I just have to let everyone know that I'm in a conference, and I think the official start will be in a few minutes. So thank you so much, everyone, for joining us. And at some point, Alvin will be the host. I may not be able to continue. Thank you so much, Homer. I witnessed when Homer was considering becoming a faculty of UP Diliman, but then he was not presented the best terms for his employment. And so he decided to continue in Genentech. And then at some point, he decided that he would just risk and create his own startup, BioAssets. And as it turns out, that was the right decision for Homer. And now I believe BioAssets is a viable company, and you have many services, then diagnostics, and you have R&D that will lead to products. So, just a little bit more on the science among all those that you identified. You indicated bacteriophages. Okay, so tell us a little bit more about that. And my second question also is, what's the price point of your diagnostics and other products that you are marketing here now, and whether these are really already developed in the Philippines, or are you also outsourcing some of them from the foreign companies and making them available here at a competitive price? Thank you, Homer.
Thank you, Dr. Concepcion. I think for the first question on bacteriophages, so the bacteriophages is a collaboration with UPLB. So we generated the whole genome sequence of their bacteriophage, which can be used which has some industrial application. I cannot disclose it here. So it has some industrial application in plants, so which we can actually protect the enhance or prolong the viability of, for example, vegetables and fruits. So, but I cannot disclose exactly what it is, but we can sit down and with the scientists in UPLB. So in terms of diagnostics, we started as a service provider. A lot of our kits are imported. So, but now the business of diagnostics is actually going up and becoming promising in the Philippines. But developing the diagnostic in the Philippines is still a lot more expensive than compared to if you import. So, but there are ways to decrease that. So that's why we are collaborating with the Industrial Technology Development Institute to develop a point of need based PCR, which is very, very close to clinical trial, and hopefully it gets approved. It goes to the FDA. But what I see from the results, it's very, very comparable with the imported PCR platforms. But this one, it does not need extraction. The problem, the challenges with the imported ones, we still have an extraction step that goes to the PCR, which takes around 3 to 4 hours. But this one will only take you less than in 30 minutes. It doesn't need extraction anymore. And I recognize the rigorous job that the researchers at ITDI have done. So hopefully, the PCR right now, you will be surprised. PCR in commercial lab is around 3,000 pesos. So hopefully with this, we can empower the farmers and veterinarians to do their test, hopefully in under 1,000 pesos. Hopefully, I cannot promise that, but let's see, because some of the other components of the kits are still imported, but I think the development is coming together already. Thank you very much, Paul.
Okay, thank you, Omer. Thank you, Giselle. Al, your turn now.
Okay, well, congratulations, Homer. You've gone a long way since our beginning conversation at the start of the pandemic. I'm glad the BIST van is already in CMU and already functioning for the last 6 or almost a year now, I think, or almost there. I was hoping that I would see you in San Francisco when I was there. But I think you're ready to shift into 3rd year, man. Pwede na magano. I'm so happy that you're doing your academy and knowledge transfer and training. So my first question is around that. I'm trying to help the mass produce our advanced manufacturing sector now in the last year or so. So I'm not sure in your production pipeline if there's any particular aspect of your product development that would entail advanced manufacturing practices. Do let me know. See if I can help in the training, modularization, and also capacity building for even as far down as our TESDA learners in order to mass produce sample takers and biosurveillance capability. It doesn't need an undergrad to be a biosurveillance person in the field. So that's one. Is that something that we can develop together? I will be with the TESDA Director General next week. And again, these are the type of advanced manufacturing type activities that we wanted to capacitate our younger learners, especially in STEM. I do have a foundation to support that, supported by Unilab Foundation, but definitely I like the academy focus, and so wanting to also address the masses that are not capable of doing college or cannot wait to go to college and earn money, but get to have some biosurveillance capability. So that's on the learning side. We'll discuss the investment and the product development, customer discovery side. There's a lot of customers for you. So I think I'll just keep it to the education side for now. And as we go through our development, and now that I'm aware of your breadth of your product development, we can discuss also your clinical validation and eventual rollout on the regulatory side as well. Yeah, so I got your input because we're developing from the NAS side also the regulatory framework that can support people like you, and of course MTech with Raul Destura. So I think I'll just focus on the I like the academy part. The product development, very exciting, but I've gone through that cycle. It's too scientific for a lot of people, but we'll just do it on a separate note. But I think I like the academy, BioAssets Academy, and what your plans, if you can have a spot for our test the learners as well. Thank you.
Thank you very much po. Right now po, the academy is focused on more on the diagnostics and also on research, how to conduct research for veterinarians and microbiologists. The manufacturing side, it will go there also. We are very excited. There are former colleagues from my previous work who are actually coming home to establish a GMP level reagent production system here in the Philippines. Actually, we're gonna do it in UPLB, which is a PESA zone now. So imagine, sabi ko nga po, as I've told you, even the smallest, littlest of water we import for our molecular biology. Right now, we have 4,000 recipes that you can pick, or we can customize it for you. Hopefully, we can do it in the next early next year, because right now we will be groundbreaking soon and establish the facility.
Okay.
Can I have a quick jump in? Holland
Go ahead,
from California.
Mr. Cheng, go ahead.
Hi, Holland.
Thank you.
Alan.
Hi, Homer. I would just actually to follow up Gisela's question about the balance between your point diagnosis versus precision diagnosis. How the line that you can draw to make it effective. So the point go ahead.
Okay. Oh, yeah. So basically, thank you very much, Holland. Holland is a collaborator. I visit him at UC Davis. So, basically, what the problem here in the Philippines is that we don't have the capability to respond at the point of need. So most because as you know, the Philippines is archipelagic, and from the southern Philippines to the central Philippines, for them to be able to get a really good diagnostic service or support, they have to send it to the major city in those region or to Manila. So it takes them a long time to get the information that they need to make a sound decision. So that's why we establish a point of need diagnostics where we can actually do the diagnostics very close to the farm, not in the farm because it will break biosecurity, but very close to the farm or very close to the vicinity. That way we can do it in an hour. We've shown it that we can do it in an hour. That way we can our veterinarians can decide whether to move the animals or not, and it has been effective, at least in some of the areas that we've done it. So the precision diagnostic is basically a little bit different. So all the samples that we get there, or all the samples that are given to us by our partners or clients, we don't stop at the yes or no, whether it's positive for ASF or negative for ASF, or positive for avian influenza or not. We continue. So because yes and no does not give you much information. It only gives you information of whether the presence or the absence of the pathogen. But if we can actually characterize at the molecular level, we can generate information that will support our diagnostic development. For example, whether this ASF or this avian influenza is still the same, whether our diagnostic will still work on these strains of ASF or avian influenza. So simple things like that. And more importantly, we want to really know to the strain level, even to the clade level, in such a way we can help our vaccine research initiative. I hope that answered your question.
It is. It's quite sad for California to lose you, but I think we can still be connected by the vaccine projects. Yeah.
I'll see you in November.
Yes.
Thank you. Yeah, Homer, I do not see any questions at the moment. So can I just ask you to share to us? Is it really the ease of doing business in the Philippines, particularly in this type of business involving a lot of science? How about the procurement you were actually mentioning earlier? How easy it is, for you to procure things that, under the Philippine, laws or regulations. Can you just share your experience to us, Homer, on this?
My very, very straight answer po is it's very hard. It's
Okay.
very hard.
So how do you go about,
Yeah, so for us as private entity, it's a little easier than academia because we don't need to deal with the PhilJeps, the bidding, and those sort of mechanisms. So we can directly buy from the manufacturers in other countries. So that way, whatever published price is there, that's the price that we buy the reagents. In academia, you buy it 3 times, right? What the challenge is, for example, I've shown you projects, 4 projects that we will have in the next 3 years, hopefully, under the Virology Institute of the Philippines program. I think the challenge that we're going to face is the speed of procurement, because the science we can do, but when will the machines, when will the reagents come? So, and the problem is, although we can do direct buying from the direct procurement from the manufacturers, we cannot do it with government money, because it has to be downloaded to the implementing agency, which is a government agency or a government institution. So right now, one of the things that we solve it is, for example, the reagent production. So for us to reduce our reliance to outside sources, and because we know we can make it, and BIST supported us for that, why don't we make it and we support the science community in the Philippines? It's small, but I think it's a good start, because what inspires me is that when I was at Genentech, there's a huge media prep. It's like a research institution already making reagents. You know what? The person who actually led that established a company named Technova. Technova is one of the biggest reagent company now that supports industry and academia in the United States. And now, using that model, why don't we do that in the Philippines? And I think it will reduce the dependence on outside sources.
Okay.
And I think other than that, I would like to. We're building collaborations now, not only with those reagents, simple chemical reagents. We're into making enzymes. Actually, a fellow PAASE member, Vic Ilag, we're actually venturing into it also. So that way we can easily support science in the Philippines.
Thank you, Homer. There are a couple of people raising their hands. Giselle, please.
Now, just to let you know, I wanted to mention Vic Ilag. More than 10 years ago, we already proposed projects to produce our own enzymes and PCR reagents in UP Los Baños. In fact, we got funding from the UPE-IDR program for this. But there seems to be a major problem in how to get the results of UPE research to the next stage of innovation and commercialization. So having BioAssets located in the PEZA SNP part of UP Los Baños is a good step of it, Homer, because then the research is based in PEZA, which allows you to procure with less difficulty and also to file IP. Okay, but also at this point, I just wanted to let you guys know that the PAASE members and also from academicians from NAST have been trying to get our views on the African swine fever vaccine that's being imported to our country to the DA-BAI. So the letter has been written and it's now with the DOST secretary to be transmitted to the DA. secretary or undersecretary. And it's very important. It's very important well, Homer, BioAssets, and others who are in the know, they have knowledge and the know-how on veterinary medicine products and concerns. Speak out as Vic and Homer and Andrea Lopez, myself, we have done this, and it should contribute to the right policies in our country. So I just wanted to say that because I think at this point, I will have to leave the meeting. So I will have to leave you with Alvin. And I hope you can show the innovation documents that we came up with in PAASE. So thank you very much, Homer, for your sharing. Thank you.
Thank you,
po.
Giselle, and all the best in your conference. So we continue. Anyway, let me just acknowledge at this point, present also in our chat this morning and of course evening in the US, our current president of PAASE, Dr. Mario Santo Domingo, our vice president and president-elect from UPLB, Dr. Gladys Completo here, and of course the members of the board who are present here, Dr. Marge Peña, and of course Dr. Al Serafica. So let us continue. I can see Al before you. Can I call on Dr. Cabellion, then later on Academician Raimundo and Dr. Completo. Dr. Cabellion, please.
Thank you, sir. Thank you, Dr. Colaba. Can you hear me?
Yes, yes.
Yes, very clear.
Yes. Oh, I enjoyed the presentation, Dr. Pantua. Thank you very much. I have a great interest in the Vaccine and Virology Institute of the Philippines from the human side. But I think I can get some correlations with regards to the animal health. So I have a question. You mentioned the nanoparticle DNA subunit for African swine flu. Just about a month ago, I read that there is a phase 3 trial in Vietnam. The vaccine was done here. My neighbor here in the United States, the National Institute of Health. I think Dr. Santo Domingo is hearing this. So they developed that vaccine, and it is in a phase 3 trial in Vietnam. So how does your DNA subparticle compare with what they are doing in Vietnam?
Yeah. Thank you very much, Dr. Silverio, Dr. Cabellion, for your questions. Just to make a disclaimer, this is a project in a collaboration of Dr. Holan Cheng, Dr. Vic Ilag, and the DOST, and our group. And just to give you an overview of the ASF vaccine development, a lot of the most advanced vaccine in the vaccine candidate in the ASF vaccinology field is the live attenuated vaccine. So the Vietnam vaccines, there are two, actually three right now that are racing. One is from a company named Navetco. The other one is an AVAC from Joint Stock Company. The other one is from the National Institute for Virus Research. So all those three are live attenuated vaccine. So the difference between those three is that the first two from AVAC, Navetco and AVAC, are gene-deleted vaccine candidates. The third vaccine is a vaccine that is cell-adapted vaccine strain. So one of the things, one of the problems with this vaccine strain is that it's not very stable. It's not proven to be the deletion is stable. What's not stable is that the challenge is that whenever you grow this virus, it induces mutations. So, and it also once you give this virus to pigs, what happens? It results in shedding. So that means it stays in the herd, and if it stays in the herd, it keeps on propagating and inducing, supporting the emergence of different mutations. The problem is not just the emergence of the mutation in that strain. The problem is that whether these mutations are introduced into the field strain, that is unknown. So what happens in China, I'm going to give you an example in China using live attenuated vaccine, is that what happened in China a year and a half ago is that the vaccine strain, which they use a genotype 1. The circulating strain in the world is genotype 2. That vaccine strain that they use, which is a live attenuated, recombined with the genotype 2, making a genotype 1, genotype 2 recombinant virus. Now, the vaccines, the live attenuated vaccines that they use against the genotype 2 does not work anymore. Okay, so that's the problem with live attenuated vaccines. So I'm not saying it's not going to work, but what I'm saying right now, we have not seen a strong data that shows that it's safe and effective. Okay, so the difference between the technology that we are using that is invented by our colleague here, Holland, is that we are going to use a nanoparticle, which is basically like a virus. Because using a virus, it will actually mimic the live attenuated, which is already adjuvanting, using the pattern, the pathogen-associated molecular patterns. So basically, we're using a virus, so it's adjuvanting already. But it's not enough. What we look at is we're going to display antigens, but subunit vaccines has been shown to be it has been very effective in preclinical studies, but it's short-lived. But in some cases, it's not also effective because it also induces the disease, because there has to be a balance between the T cell response and the humoral response. So this is a very, very exciting time for science. That's why the scientists in the name of Holland and Vic is that what we thought is, why don't we put both T cell epitopes, the T cell factors, and the humoral factors, such that we have a virus that we now have a virus, like a virus, that will mimic the same immune response as the live attenuated, but without the danger of being of replicating or in shedding or recombining with the wild type strain. So that's the beauty. But now it's just an idea. It's just an idea. We need to do the experiment, and we are glad that our government is betting on us. Thank you, Bob.
That's great. Thank you, Homer. Thank you, Dr. Cabellion. Yeah, Academician Raimundo.
Yeah, first of all, I'd like to applaud Dr. Homer for the guts and courage to give up his job in Genentech and come up with a local BioAssets and its novel goals for R&D in the and science in the Philippines. I was wondering, while you were talking about your diagnostics, there are plenty of PCR-based diagnostic research done in at least UPLB. I, for one, with my students, have come up with a PCR-based diagnostic for the bacterial pathogen, and there are lots of them in the so-called shelf research results, because there were no more funding, so we were not able to pursue it. And there are still others like salmonella-based PCR diagnostic for salmonella, etc. I wonder what the BioAssets can do something about that, maybe through its academy and our students. Thank you.
Yes, ma'am. Thank you very much, Academician Raimundo. Classmate ko po si Marvin.
Oh, really?
No elementary. Anyway, thank you very much for your question. It's a very, very interesting question, because one of the thrust of BioAssets is technology commercialization. I think based on my background in the US, we don't always have to do the R&D, because a lot of times the R&D is very expensive. That's why with researchers like you, we want to partner with you and look to see where are you and how we can actually move those technologies into the market. We can co-develop it, or we can, if you are ready, we can actually support your trials or some sort of that, or we can train your people so we can continue with your research. But definitely po, we can support, we can talk about technology commercialization in the Philippines.
That would be great. Thanks, Summer.
Thank you. Thank you, academician Sean. Dr. Completo, Gladys, your turn.
Hi, good morning.
Good morning,
everyone,
Paul.
and good evening. Congratulations, Dr. Homer Pantua.
Thank you,
So I'm very interested about your intention of producing reagents needed here in the Philippines. So what type of reagents are you starting to produce now, or are you just planning to start doing it?
So it's in the plan po. We are actually groundbreaking in the next sometime in October. We got approved last week with our lease with UPLB. So that's very exciting po. We have the plans and layouts. We have the list. Whatever you need po, we will customize for you.
Okay, that's great, because if you want collaboration as well, because we're trained synthetic chemists here, and then we're just, and I'm based in UPLB, so we can meet, and just let me know and you can contact me po.
Yeah, thank you very much po. But basically it will be research reagents, to the simplest water, to PBS, to TV,
Yes.
to buffers, DMEM, everything po. The plates that are tested for everything, tested. For example, water, PBS tested for low endotoxin, something like that. Hopefully enzymes will be able to do that too, in collaboration.
That's nice. We're interested to collaborate with you, please.
Thank you very much po, Dr.
Thank you.
Yeah, thank you, Gladys. Maybe Homer, you can we can call on yung mga universities doing research, that would need reagents, and then buuin na natin yung di ba, effort, and so because I'm sure, because we are separated, sabi mo nga, it's so difficult in the provinces and the regions, parang ganon, to access these things, or even within the country, within the Philippines. So kailangan na natin, i-disperse na natin yung development para sabi mo nga, that's why you brought the mobile lab in CMU or in Mindanao, di ba? To get more easy access to this. And I think we have to do that, across, the different islands. So thank you, Gladys. I hope you can help initiate that move, and then, but let's do that for the entire, country.
Yes.
Doc M, before sa'yo, I'll go back to Dr. Serafica. You're still raising.
I'm sorry, I wanted I was trying to leave out the technical, but since you were already brought into the discussion, clearly you're addressing a user need, Homer. I was actually contacted by GenScript about a year ago about the potential for producing some of these reagents over here. And I was looking for a partner downloader because I don't have the bandwidth to do it. Maybe we can revisit that, because we visited, I was part of the ASEAN Diagnostic Hub for the last 3 years in Singapore, Philippines, with PCHRD. And that was really more preparation for human diagnostics. But we visited Biopolis, so I had a view of some of the startups that's coming out of Biopolis in Singapore. And you're actually pretty much along the same line. And clearly, I think it's time to plug in. I'm putting my PAASE board hat and the program for next year is to have a stakeholders meeting on your area on life sciences, both human and animal, Gladys, for have a program. Homer, when are you coming home? When are you physically here?
I'm in the Philippines po.
Ayun na, in the Philippines. Okay, let's get together then. That's one part, and then so we can discuss more the composition. But definitely a program, Alvin, for PAASE and Gladys on life sciences that would include both agro and agri, and invite DA and all of the other collaborators and the SUCs that have interest. I think I'll invite na rin while he's here, Rolly, the engineer Sevilla, please do come, because part of the ecosystem stakeholders are the risk capital and the entrepreneurs that we're trying to assemble, not just government, industry, and academia anymore. We need more entrepreneurs and risk capital to be involved. I was an entrepreneur for 20 years in the US before I came back here as a Balik scientist almost 10 years ago now. We've been trying to increase the number of startups and venture capital involved. It has started already on the fintech, ICT, and artificial intelligence and data science, but life sciences has been slow. Only MTech and BioAssets are the only companies that I know that have scale right now, that have reached over 10 to 20 to 100 million in terms of potential revenue. So I think we have examples, but with 2 in line and another 3, hopefully the metabolomics discussion that we had with Gladys and UC Davis professor Carlito Librilla, and that's my input to Holland. Cheng, do come here in July, because I'll be inviting. I was with Carlito Librilla of chemistry, has 6 LCMS machines running all the time in Davis, and looking for more things to analyze, Homer. So I think we ought to consider this cross-border relationships with UC Davis. I visited Citrus also with the Dato Banato Institute. So I wanted to be able to build this ecosystem with you being in the center and what you're doing, and capacitate what you've started. Thank you.
Thank you. Thank you.
Thank you.
Let's start to Gladys, to scale this up. No, I think we have to really identify institutions who are already ready, and get involved in this initiative, and let's form like a consortium. We had to do that that way. I mean, we cannot be doing it in individual institutions, because the problems are just too many. We need more people to help, the ideas, resources. I think that should be where we should be directing our efforts to. And I'm sure, but the critical thing there is to identify who are ready in this level of work. I think, and I'm sure there are many across the country. So let's not it's always think from north to the south. Let's get them involved now. It's very important that they at least they have the idea, because they will. I'm sure they have the resources to make this happen. Now, Gladys, before you, I think you're raising again, but let me go to Dr. M
Sorry,
first.
no, I'm done.
Dr. M.
I'm okay.
Go ahead.
Al, please contact me. Sorry, can I just remind Al to contact me when he's here in UPLB, okay?
Sure. Okay.
So that we can
All
start.
right. Doc M, the floor is yours now. Thank you.
Thank you very much, Lava. Good morning. The College of Veterinary Medicine is so proud of you for having an alumnus that has well accomplished this field and all. I'm very much interested about itong African swine fever, itong vaccination. You mentioned that the virus is very unstable. That's true, and it keeps on mutating, and there are always new strains emerging. So my question is, can we ever have a safe and effective ASF vaccine for use in the country? And if so, kailan pa, gaano katagal maghihintay ang swine industry? Kasi alam mo naman, still it's a big problem, itong African swine fever. So can we ever have a safe and effective African swine fever? I'm asking you this because, as I states, they're not even planning to use or to undertake vaccination against African swine fever.
Yeah, thank you very much, Dr. Maala. He's one of my favorite professors in Vetmed. Anatomy. Yeah. So, but anyway, the first before I answer the question, right now I just came from an African swine fever workshop. It's a meeting of 40 scientists across the globe, looking at solutions for African swine fever, basically focused on vaccine development. And the summary of the meeting is that there is still no safe and effective vaccine. That's the summary of the meeting, Paul. And to answer your question, Paul, with the flow of information that we get from African swine fever virus biology, the biosecurity, I think there will be a safe and effective vaccine. I cannot say when, because right now, even the subunits has the live attenuated has its negatives. Even the subunits has negatives too. So that's why you have to learn more in such a way that we can actually generate one that is safe. Effective, yeah, it could be effective for 1 cycle, 2 cycle, 3 cycles, and then after that, it will give you a problem. For example, the live attenuated. But the subunit, we have to prove that it's long-lived also. That we have to prove that the subunit will not induce antibody-dependent enhancement, which is being seen right now in the absence of T-cell response. So you need to have a balance in those two. Right now, we've been learning that the African swine fever, actually what's happening is that pigs, when they have African swine fever, they have sudden death without any clinical signs. And what we're thinking is that, just to give you a background, before I left Genentech, I was working on sepsis for the last 3 years. And what I'm suggesting is that with the other researcher is that what's happening right now is basically sepsis. Because what happens is that the virus infects macrophages, and the macrophages will actually induce a cytokine storm. For example, a lot of cytokines, IL-1 and IL-8 and other cytokines. When it induces a cytokine storm, it will actually induce sepsis. So that's why there's a sudden death of the animals. So those are one of the things that the industry is that the research the researchers are looking right now. Actually, I'm very excited. I've talked to one. We'll be collaborating in the next months, looking at that question. So, but yes, the answer, yes, there will be. When, I don't know.
Thank you, Homer. Thank you.
We have to do a very, very thorough clinical trial. It can't be few animals. It has to be a large-scale, massive clinical trial to prove efficacy and safety.
Okay.
Thank you.
Thank you, Doc.
Thank you. Thank you, Academician Maala. Yeah, time check, we're 10 minutes. Actually, we want to close this at 10 o'clock. Pero you just mentioned Homer, clinical testing. Ito gusto ko ito itanong, no, kasi we were involved in development of a respirator, no, during the pandemic. So nagawa namin yung machine, no. Ito yung ito, I'll knows this, no. So nagawa namin yung machine, it passed all the mechanical testing, ready na siya gamitin. Kaya lang kailangan daw mag-move kami to the next phase muna kasi it will have to be to humans. Yung clinical animal testing. So na-stuck kami dito kasi sa Pilipinas wala. Now we learn from St. Luke's Medical Center, meron sila animal testing facility. Kaya lang if you are going to use animal mga subjects, no, like I think we were thinking like 20 pigs ang kailangan namin or what. Walang facility sa kailangan kasi i-quarantine pa pala yun, i-prepare pala yun, no, before they are subjected to the animal testing. So UPLB has that yun nga yung mga logistics and everything. How can you help suggest going in that stage of testing machines or na kailangan pala, akala kasi namin didiretso na kami sa human testing Hindi pa pala pwede. Kailangan pala kasi wala kaming alam Diba? We are mechanical engineers, we are, sabi na hindi pa pwede, kailangan muna kayo mag-animal testing. Eh yun, hanggang ngayon walang nangyayari. So, Homer, can you comment on that? How do we get to that stage? Before I go back to Al. Sige, go ahead.
Of course. Opo, it's very important po. All medical devices that involves human use, vaccines, therapeutic, has to go to preclinical studies in an animal model po. Lahat, everything. And one of the best clinical model is the one close to us, our monkeys. We have a lot of the monkeys that is being used globally, the macaques, the cynomolgus macaque. It's being used globally in many contract research organizations. The nearest to us is Singapore. A lot of them are in the United States. The problem is we don't have it here. It's not set up in the Philippines. Right now, there is some initiative to set that up. Actually, we are very interested in that. Lab animal medicine towards a contract research organization. Basically looking at very small things, PKPD of antivirals, therapeutics and vaccines. PKPD lang po. But hopefully, I think it's doable. The problem, the challenge is it's very expensive to do preclinical studies. Again, for example, if you're going to do it in non-human primates, I'm not a lab animal medicine expert, but we have a consultant right now who is also a Filipino who graduated from the College of Veterinary Medicine, who is now the head veterinarian at LabCorp. So he's helping us. It's definitely doable po. And I think if we do it here in the Philippines, if there are some investment, it's going to be cheaper, because right now I'm going to tell you, you're going to be surprised. We have so many cynomolgus macaque here in the Philippines. When those macaque goes to the United States and do your medical testing device, monkeys are worth $40,000.
Wow.
Monkeys of 2 years old, the age is 2 years old. When it becomes 3 years old, 4 years old, it can be $50,000. When it was $5,000 like 5 years ago. So now it's at least $40,000 for 2 years old. But imagine, I am posing it to Sir Al, if there are investors who can help, we can definitely do it here in the Philippines. Okay. We have the monkeys already. But of course,
fully agree.
We cannot use wild monkeys. We have the monkeys. We can only use F1s to F2s.
Yeah, we have to breed them. Actually, Homer, there was a facility, according to academician Padolina, about 20 years ago, because I was trying to also bring the clinical, that closed up 15, 20 years ago. But also I've been helping one of your other COBIST awardees is JV Solesta, who was coming up with 3D printed human skin equivalent for some of the pharmaceutical. There is a biochip capability of one of our PAASE members, Dan Tagle, that was developed in Harvard Wyss Institute for potential cell-to-cell interactions of bioactive agents. I don't know yet the eventual gateway kasi Homer and Alvin is our regulatory body. Part of the reason that you couldn't get it into humans is because you don't have any investigational device exemption, Alvin. We should have gotten that. In the US, they will issue you under an EUA emergency authorization similar to the diagnostics. Under a COVID emergency, you should have been allowed to do that. But unfortunately, our FDA still needs to also come up to terms in terms of the modern era of FAST. So that's why the regulatory of one of our special projects with NAST right now is to really convene the regulatory bodies as well as the practitioners. And maybe you might want to show up on that particular roundtable discussion to represent the vet area, Homer. We're going to convene that probably we're projecting it siguro sometime either December or January, probably January na, under the NAST for the regulatory. Kasi we're trying to do clinical advancements. And again, people tend to forget that animals are also needing clinical evaluations and validation and verification. So I think sabay-sabay na lang natin lahat, Alvin. So, but thank you very much. I mean, there are solutions, but I'm glad that you're part of the ecosystem, Homer. Thank you. We'll do a lot of stuff. Thank you.
Thank you, Homer. Definitely, you have more engagement apart from your own company, and to help the STI ecosystem in the country. So of course, we're only 3 minutes away from closing this chat. Very ang dami pang pwedeng i-discuss, but I'm sure this will not be the first time that we're going to bring Homer here in this forum. So I would like, before we finally end, and of course, thank Homer, let me just show you here. You can see in the slide the subsequent series of our Fireside chat. Pakilaki-laki nga, Feb. I cannot read it, sorry. But of course, we have Gobet, Dr. Advincula, Dr. Turingan, Ralph Turingan, Vanessa Yingling, Mark Lawrence Cruz, and Dr. De Jos in our future Fireside chats. So we will keep you informed, of this, events. Of course, next Friday na ba ito? Feb, si Dr. Govet Advincula on materials, manufacturing, and artificial intelligence. So anyway, what's next, Feb? These are some of the initiatives of the National Innovation Council that PAASE would like to support. So through PAASE, there are already forms 1, 2, and 3 that you can check on our website for grant proposals. It's not very clear yet what are the guidelines and criteria for this under the NIC. We have the paper on Philippine S&T innovation, if you want to also access that, and on barriers to innovation and propose solutions if you may want to raise some of the ideas there. PAASE can facilitate this, as we try to build a more, realistic innovation policy, for the country. So, is there any other, Feb? Next, none? Okay. So, we would really like to thank and express our great appreciation to Dr. Homer Pantua. Excellent talk, excellent sharing of your experience, of setting up, a company in the Philippines, and more importantly, serving, the Filipino people and the country, primarily in the animal, industry. I'm sure your experience will inspire and motivate more innovators to set up their companies, startups, and similar undertaking in the Philippines. And I think this not only for Filipinos abroad coming to to this country, but I think more importantly our own innovators and scientists who can join together to set up a business in the Philippines. I think you need to shepherd, to guide these people because you have the real experience. Also accessing BIST, for example, I think that's a good idea. We have not seen success stories on that, and I think Homer has shown that it can be done and it is viable to seek like a loan from the government to the DOST. You can share the viability of that particular program, Homer, that it works and it can help set up even small things to start with in this country. So again, would like to thank PAASE for this. Mario, are you there, Mario? Santo Domingo, our president, Marge, Peña, I think she's here, and of course Gladys, you will be our incoming president. There's just so much things that we can do, with PAASE and our major, collaborators in the private sector. So let's capitalize on that. I think we are now ready, I think, to really embark in a more realistic, initiatives that can help. But let us help the entire country and not only, focused on especially here in the National Capital Region or in Calabar Zone. There are just so much talents around and resources also that are available elsewhere. Let's bring them into our fold, into our discussions. So, and hopefully we can realize in a very short period our aspirations, for the country. So, thank you again so much, Homer, for your time. Thank you very much. Appreciate your sharing. And we hope that, I know you mentioned that you are here every 2 months, every other 2 months between Philippines and the US. So I hope hindi ka magsasawa sa mga request to
Uwi na yan.
share.
Dito na yan.
Ito na yan.
Thank you so much. Of course,
Hindi na alis yan.
Yeah, I would like also to acknowledge the presence here of National Scientist Dolores Ramirez of the National Academy Science and Technology, and of course our Tito Aliga and our mga regular viewers here. So thank you very much, Homer. It's already 10.
Thank you, Homer.
Photo
Thank
up
you,
daw.
Alvin.
Thank you.
Photo up daw po, Dr.
Open your
Alvin.
camera.
Sige, please, kindly
I-
open your cameras.
As I always say, proof of concept. You
Proof
can
of
never
face.
beat that.
Proof of life.
Proof of
Proof of
concept.
life, Tito.
Yeah, sabi
Buhay
ko,
pa tayo.
from the ground to the cloud to the ground eh. Importante yun. Thank you, Homer.
Thank you.
Okay na?
Thanks,
Sige.
Homer. Very interesting.
Homer, let's
Thank you.
bring that to the Visayas, to Mindanao. You're
Yes.
in Mindanao. So thank you so much for your, time.
Thank you
we
very
look
much,
forward to
Tito.
your more active engagement. Thank you, everyone.
Thank you.
Good day, and of
Thank
course,
you.
good evening over in the US. Thank you so much. Have a great day and
Great
stay
day.
safe, everyone.
Yep.
Bye-bye.
Thank you.
I'm here in Taichung now, in
Where you
Taiwan.
are? Oh my goodness.
Wow.
Yeah.
Bye-bye.
International
guys.
conference.
Thank you.
Fed,
Bye-bye.
next
Bye.
time,
So much.
Thank you, Fed.
i-post natin
Fed
yung
Clares,
exams.
behind the logistics of this fireside chat. Thank you very much, Fed. Bye-bye, everyone.
Bye-bye.
Homer, I hope to see you
Thank you po.
sometime.
Thank you.
Bye-bye. Thank you, National Scientist Ramirez. Bye-bye.